Welcome to Pharmacology Insights, where we break down complex pharmacology into simple, easy-to-understand concepts. What if, instead of just blocking a cancer-driving protein, you could make the cancer cell destroy it from the inside? That's not science fiction. That's exactly what Vepdegestrant does. And as of May 1, 2026, the FDA has officially approved it for breast cancer. In this video, we'll break down how this drug works at the molecular level, why it's genuinely different from anything that came before it, and what the clinical trial data actually tells us. Before we talk about the drug, we need to talk about the target. Breast cancer isn't one disease. It's a family of diseases. The subtype we're focusing on today is called ER-positive, HER2-negative breast cancer. This is the most common subtype of breast cancer, accounting for roughly 70 to 80% of all cases. So what does ER-positive mean? ER stands for the estrogen receptor. Specifically, estrogen receptor alpha, encoded by a gene called ESR1. In normal breast tissue, estrogen binds to this receptor. The receptor enters the cell nucleus and switches on genes that drive cell growth. That's normal physiology. But in ER-positive breast cancer, that process gets hijacked. Cancer cells become addicted to estrogen signaling. It drives their proliferation, their survival, and their spread. This is why endocrine therapy has been the cornerstone of ER-positive breast cancer treatment for decades. We've had tamoxifen, which binds to the estrogen receptor and blocks it. The receptor is still there, just prevented from doing its job. Aromatase inhibitors like letrozole take a different approach entirely, cutting off the estrogen supply before it even reaches the receptor. Then came fulvestrant, which goes one step further than tamoxifen. It not only blocks the receptor but also destabilizes it, causing the cell to partially break it down. And more recently, CDK4/6 inhibitors like palbociclib and ribociclib, which block cell division downstream of the receptor altogether. Now the standard first-line combination partner. But despite all of that, cancers find ways to escape. In advanced or metastatic ER-positive breast cancer, resistance to endocrine therapy is almost inevitable. Here's how it works. Under the selective pressure of endocrine therapy, particularly aromatase inhibitors, cancer cells can acquire mutations in ESR1. The two most common variants, Y537S and D538G, lock the estrogen receptor into a permanently active state. The receptor behaves as if estrogen is always present, even when there's none. Drugs that lower estrogen levels? Useless. Drugs that compete with estrogen to block the receptor? Far less effective. ESR1 mutations are detected in up to 40% of patients with endocrine refractory metastatic breast cancer. This is a massive clinical problem. So what do? You need a drug that doesn't just block the receptor. You need one that eliminates it entirely. Even the mutant versions. Enter Vepdegestrant. This is where things get genuinely exciting. Vepdegestrant belongs to a completely new class of drugs called PROTACs. That's short for proteolysis targeting chimeras. And I want you to understand this concept, because it represents one of the biggest paradigm shifts in drug design in decades. A PROTAC molecule is bifunctional, meaning it has two distinct binding ends, connected by a chemical linker. One end binds to the target protein, in this case, the estrogen receptor. The other end binds to an E3 ubiquitin ligase, a cellular enzyme whose job is to tag proteins for destruction. When Vepdegestrant brings these two proteins together, the E3 ligase attaches small ubiquitin tags to the estrogen receptor. This is the cell's own molecular signal for disposal. Once the receptor is tagged, vepdegestrant attaches. Its job is already done. The tagged receptor is then recognized by the proteasome and destroyed, while vepdegestrant is free to recruit another E3 ligase and destroy another estrogen receptor molecule. This is called catalytic recycling. One drug molecule can destroy multiple target proteins in sequence, something no traditional drug can do. A conventional drug has to stay bound to its target continuously to keep it blocked. The moment it lets go, the target bounces back. Vepdegestrant just needs to trigger the event. Then it's already gone to cause the next one. In laboratory studies, vepdegestrant achieved up to 95 to 97% degradation of the estrogen receptor. Near total elimination. And because vepdegestrant works by removing the receptor, rather than competing at its binding site, it's active against both wild-type and ESR1 mutant receptors, including those Y537S and D538G variants. The proteasome doesn't care what mutations the receptor carries. Tag it with ubiquitin, and it's gone. Now let's talk real-world evidence. Does this work in patients? For a specific patient population, the answer is a compelling yes. VERITAC 2 was a landmark Phase 3 study. This is the first Phase 3 trial of any PROTAC drug ever conducted. The trial enrolled 624 patients with ER-positive, HER2-negative advanced or metastatic breast cancer, who had already progressed on at least one CDK4/6 inhibitor, plus endocrine therapy. Patients were randomized 1:1 to receive either vepdegestrant, 200 mg orally once daily, or fulvestrant, 500 mg by intramuscular injection, once monthly. The primary endpoint was progression-free survival, analyzed separately in the ESR1 mutant subgroup and in the overall population. In the ESR1 mutant subgroup of 270 patients, vepdegestrant achieved a median progression-free survival of 5.0 months, compared to 2.1 months with fulvestrant. That's a 43% reduction in the risk of progression or death, with a hazard ratio of 0.57. The objective response rate, the proportion of patients whose tumors actually shrank, was 19% with vepdegestrant versus 4% with fulvestrant in the ESR1 mutant group. That's nearly a five-fold difference. The FDA approved vepdegestrant specifically for ESR1 mutant tumors. And the trial data shows exactly why. In patients without that mutation, vepdegestrant and fulvestrant performed virtually identically. 3.7 months versus 3.6 months. No meaningful difference. The mutation is what unlocks the benefit. So, what does vepdegestrant actually bring to the table? First, it's a pill. That sounds simple, but it's clinically meaningful. Fulvestrant is given as 2 painful intramuscular injections per visit, once a month, every month. Patients consistently describe this as one of the most dreaded aspects of their treatment. Vepdegestrant is taken at home, once daily, with food. No injections, no clinic visits for administration. Better quality of life. A genuine practical win. Second, more complete receptor destruction. Fulvestrant is itself a selective estrogen receptor degrader, but it works indirectly, triggering conformational changes in the receptor that eventually lead to partial degradation, reaching roughly 40 to 50% at its optimal dose. Vepdegestrant directly recruits the cellular degradation machinery, achieving up to 95 to 97% elimination. That's not incremental improvement. That's a mechanistically superior approach. Third, activity against resistant ESR1 mutations. Because vepdegestrant degrades the receptor rather than competing at its ligand binding site, the mutations that blunt fulvestrant's effectiveness are largely irrelevant to vepdegestrant's mechanism. The VERITAC-2 data confirm it. The ESR1 mutant patients, the ones most likely to have failed prior endocrine therapy, are precisely where vepdegestrant showed its strongest results. Fourth, catalytic recycling. One vepdegestrant molecule can destroy multiple receptor molecules in sequence. After each degradation event, vepdegestrant is released intact and cycles back to work. Greater target knockdown, potentially at lower drug concentrations. On safety, vepdegestrant was generally well tolerated. Serious side effects were slightly more common than with fulvestrant. But treatment-related deaths were absent, and fewer than 3% of patients stopped treatment due to side effects. Science demands honesty. Here's what vepdegestrant does not yet tell us. Overall survival data are not mature. We know vepdegestrant delays progression in ESR1 mutant disease. We don't yet know if it extends lives. Progression-free survival is not survival. That distinction matters. The benefit is specific to ESR1 mutant tumors. In the unselected population, vepdegestrant did not significantly outperform fulvestrant. ESR1 mutation testing, via liquid biopsy or tumor tissue, is mandatory before prescribing. A median progression-free survival of 5 months is meaningful compared to 2.1 months, but it's still modest in absolute terms. Combination strategies will likely be needed for deeper, more durable responses. And long-term safety and resistance patterns for PROTACs as a class are still being mapped. This is the first approved PROTAC. We will learn more about novel toxicities, about how tumors escape this mechanism, as clinical experience accumulates. And that's it for today's pharmacology video. If you enjoyed this video, please give it a thumbs up and subscribe to the channel for more pharmacology content.